Hello and welcome to your lesson on experiment results. In this lesson, we're going to be looking at the experiment in terms of the data you collect. We're going to look at how you might do some graphs, understand the different types of data you can obtain. We're going to look at different errors, and then we're going to finish off by looking at how you would form a conclusion, and also having a quick look at that example of the experiment setup to look at the effect of light on the direction of plant growth. So the experiment, what's the point of it? It is carried out to gather data and it should be repeated numerous times. Why? Because it's going to make it more accurate. And the results should always be recorded and put in a table, a bar chart or a graph, etc. based on your needs. In terms of graphing, it's a really important skill. So you need to consider things like, is your graph salty? Does it have scale? Does it have the axis? Does it have label? And does it have titles? So the scale, the accurate horizontal and vertical scale shown, it should have the independent variable on the horizontal. It should have the dependent variable on the vertical. In terms of the axes, the scale should be clearly shown. The value should increase by the same amount each time, and they should both start at 0. In terms of labeling, and this is something people always forget to do, you should label them. Okay, so the horizontal could be time in hours or temperature in degrees Celsius, and then the title should explain the purpose of the graph, and be aware of the nature of your graph. Is it a curve or does it require a line of best fit? And again, you can see it all explained over here as well. Make sure you always remember your unit when you're labeling the different axes. Okay, so as we move along then, what are the results? The results are the data produced from the experiment. For example, the measurements are the information gathered. Now, the data you obtain should be obtained from what we call replicates, which are repeats of an experiment which help to guarantee statistical reliability. If you do something once and you get a certain set of results, that is not enough. It's not going to give you any sort of statistical reliability. If you repeat it again and again and again and again and keep getting the same data, that then provides a bit of statistical reliability and puts a bit more weight behind your findings. Now, in terms of the data itself, there's lots of different forms. So you can have qualitative or quantitative. So qualitative data is based on descriptions of the findings. So for example, color changes, smells, characteristics or traits. It's something you can describe, whereas quantitative data is numerical based measurements. It is an actual numbers based game. So you're looking at a temperature change of 20 to 50 degrees. That's quantitative data. The mass changes from 100 grams to 120 grams. That is quantitative data. So that's one type of data. Another will be the idea of primary and secondary. And this will matter in terms of, you have to be very clear, are you obtaining these results or are you getting these results from somewhere else? So primary data is gathered firsthand during investigations. So I measured the temperature change from 20 degrees to 50 degrees using a thermometer, whereas secondary data is gathered by someone else. So you could get it from a scientific paper that provides information on genetic disorders from a lab-based experiment in a university. You didn't do that. It's still very useful data, but it's not your data, so therefore it's secondary data. Now, when we're gathering data, it's very important we understand that there are always going to be errors. So random errors are a result of chance and they do not repeat again and again and again, whereas systematic errors will be repeated over and over again, and they're repeated differences between the value obtained and the true value. Now, in terms of their impact and how to reduce them, we need to be very aware of what's causing them. So random errors could be an individual human error on one particular experiment, whereas systematic errors are like the equipment isn't working correctly. So if we look at this, random errors will impact and reduce when you repeat the experiment again and again and again and find averages, whereas systematic errors are due to problems with the equipment or how the equipment is being used, and therefore you need to ensure that you're using the equipment correctly and you're like setting the balance to 0 each time, or else you're going to keep getting the same error over and over and over again. So to reduce systematic errors, you have to take care while using the equipment to ensure that it's working correctly, it's calibrated, and this will help then reduce those systematic errors. And even just knowing that there are errors is important, okay? Because this is going to affect the accuracy and precision of your results. So being aware that there might be errors and acknowledging this can also be very important in terms of how you form your conclusions. So in terms of accuracy, this is a measure of how close your results are to the true value, whereas precision is a measure of how close each set of measurements is to each other. So if we look at this, if we pretend this is a target here, this is an accurate and precise experiment. Here we have a low accuracy but high precision, because accurate, we want as close to the middle as possible, but precision, all the data points are close together. Here we have a decently accurate, so it's a high accuracy but low precision because all the data points are far away from each other. And here we just have a really poor experiment that has low accuracy and low precision because they're not close to the center of the target and the data points are very, very far away from each other. So you have to understand the difference between accuracy and precision. So accuracy, close to the true value; precision, data points that are close together, and then you want to get results that are both. Okay, so it is important to always be aware of accuracy and precision when you're carrying out your experiment, as this will have a significant effect on your findings. And then once scientists have their data, they need to critically analyze data to identify patterns and relationships, identify any potential anomalous observations or unexpected observations, and then form a conclusion and report their findings. So let's look at an example here. So here's the results of the experiment we showed in the previous lesson. So we're looking at the effect of the direction of light on plant growth as shown below. So what we did was we ensured that there was only light coming from one direction here and one direction here. And then we had a control here in the middle, which had light as you would be expected. And what we find is, if you were to consider the hypothesis for the experiment, you would hypothesis if the direction of light changes, then the plant will grow in the direction it is receiving light because it needs it for photosynthesis. That'll be your if then because way to form a hypothesis. And what you'd find is that is most definitely the finding. So here the light was coming from over to the right, the plant grew to the right. Here it was to the left, so it grew to the left. In terms of the control experiment is the plant that was getting light in its normal direction, so it just grew as per normal. In terms of the actual variables, control variables here would have been the duration of light exposure, the amount of water being given to each plant, the type of soil, loads and loads of different things. The independent variable here would be the direction you're receiving light from. The dependent variable then will be the direction the plant grew. So here we can see very much so that the hypothesis was correct. The plant did grow in the direction that it was receiving light. In terms of accuracy and precision of this is only showing 3 plants, therefore it's not going to be very statistically reliable. We don't know how accurate it is because we don't know if somebody made sure that the control variables were constant throughout. So we have no idea how accurate or precise this experiment was, and we also don't know how many times it was done. So to make sure this was really, really well done, this should be repeated many, 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 many times, and then averages are like, you're finding, does this result happen over and over again? And if that's the case, then you can say it's accurate, it's precise, and has statistical reliability. So if you were to look at this experiment and then do what we said a minute ago, whereby you're looking at it and you're trying to form a conclusion, what does that mean? So in your conclusion, you'll state whether or not your hypothesis was correct and accurate. This is done after you have analysed your data. So in this case, the data was the plants grew in the direction they received light from. So then straight away, you're going to say, my hypothesis was correct. The plants grew in the direction that they received light from. You will also then to try explain your results. So in this case, they grew in this direction to receive as much light as possible to ensure that they could photosynthesize and survive. And then when you're forming a conclusion, you also must consider potential errors within the experiment to be aware of things like causation versus correlation and whether or not they're impacting the accuracy of your conclusion. So here you'd say there may have been errors in terms of the duration of light or whatever errors you may have made. You just acknowledge them, because then that makes you aware of this idea of causation and correlation as you form your conclusion. And just to be very, very clear on this, causation and correlation is as follows. Causation is whereby a change in one variable is due to a change in another variable, whereas correlation is whereby there's a link between the two, but they aren't directly affecting each other. So dry, hot, sunny weather causes you to want to eat lots of ice cream, and it can cause you to have sunburn. Ice cream and sunburn are correlated. They're not causing each other. Ice cream doesn't cause you to have sunburn. Sunburn doesn't cause you to have ice cream. The cause is the sun. The correlation is hot, dry, sunny weather. These two things both happen quite regularly. So there is a very big difference, and you need to be aware of that when you're forming your conclusion. And then finally, a little guide here to help you form your conclusion. Think care, so clear aim, restate what you were testing in the experiment, analyze your results, so summarize the main findings and patterns of your data, relate to hypothesis by comparing your results to your hypothesis and connect the independent and dependent variables, and then E, explain why, use scientific reason to explain your results. So in this experiment, we were testing to see the effect of the direction of light on the effect of plant growth. We found that the plants grew in the direction that they received light from. This confirmed our hypothesis whereby the independent variable of direction of light caused the plants to grow in the direction that it was coming from, which was the dependent variable. And then you'd use scientific reasoning, so you go into detail as to this is because the plants needed light to carry out photosynthesis, which is required for them to survive. So think care every time you're doing a conclusion. Finally then, a couple of questions to finish off. What is the difference between qualitative and quantitative data? What type of results in terms of accuracy and precision does this image represent? And what is causation? Pause the video, jot down your answers, come back and I'll give you the right answers. Okay, so the difference between qualitative and quantitative data. Qualitative is based on descriptions of the findings, while quantitative is numerical based. That image there showed low accuracy but high precision because the data points were close together but not near the center of the target. And causation is whereby a change in one variable is due to a change in another variable.
If you like this video, you'll love our notes. You can get some for free in the link in the description of this video.